Thank you so much for everyone at Café Sci, and thank you, Armand, for that introduction. That could be a conversation on its own too. And absolutely, my path through science has been very unpredictable. And I think that's one of the things I like about science is we really can follow our interests as long as there's some element of curiosity. That's what I really fell in love about science, but we can have that conversation later. But today, I'm going to be sharing with you a little bit about my research interest at San Jose State, particularly in what Armand said, like my recent venue into this field of cardiac regeneration. So I'm going to go ahead and start sharing my slides now. Okay, so hopefully everyone can see that. All right. So the title of my talk today is Lessons from Newborns, Neurohormonal Signals Limiting Mammalian Heart Regeneration. Again, I am a relatively new assistant professor in the Department of Biological Sciences at San Jose State University. I'm happy to talk about career as well as science, so please feel free to reach out, okay? But today's topic is going to be about heart regeneration, and to really engage you in why that's important is just to remind you, and I'm sure it's no surprise to a lot of you, that heart disease remains a leading cause of death in the United States. In fact, according to the CDC, 1 in 4 deaths in the United States are because of heart disease. So what is heart disease and how does it arise? Well, a lot of times, due to narrowing or blockage of major blood vessels in the heart, right, we might deprive downstream tissues of blood and oxygen and nutrients, right? This is what we call a heart attack. So this area that's kind of shaded gray is being deprived of oxygen, nutrients, and eventually that tissue dies, right? When this tissue dies, the rest of the heart has to figure out what to do. The heart's a very critical organ. It needs to pump and circulate blood, oxygen, nutrients to the body. So there's remodeling that occurs in the heart. This remodeling can be pathological, eventually leading to heart disease and heart failure, right? So to engage everyone, what I'd like to do, and just a warning, is I do have some questions prepared in terms of polls, just to survey what people think about, and to just get your mind thinking about the heart and some of the topics that we're going to talk about today. So the first question that I have is this one. So this is just to realize that the heart is actually composed of cells, right? And not all the cells in the heart are the same thing. We talked a little bit about in the previous diagram, there are blood vessels in there, right? So here's a question I have for you. Which cell type in the heart is contractile? Is it A, cardiomyocytes, B, fibroblasts, C, endothelial cells, or D, stem cells? So let's take about a minute or so, or I'll get a sense when people are done, but let's take no longer than a minute on this one. So what do you think? What are the contractile cells in the heart? What makes the heart beat? Wow. Getting good participation on this one. So let's take no more than 20 seconds on this, okay? 20 more seconds. So if you want to chime in, let's do so in the next 10 seconds or so. Okay. So let's go ahead and end the poll. I'm going to go ahead and share the results. So everyone should see kind of what I see, and if not, I can drag the results over here. But about 71% of you said cardiomyocytes, okay? And that is correct, right? So which type of cell in the heart is contractile? Cardio, heart, myocytes, muscle, right? Contractile cells. Fibroblasts are actually more with the connective tissues of the heart. The endothelial cells line the blood vessels. And we're familiar with what stem cells are, and we'll bring this up slightly in a bit, okay? So I'm going to stop sharing. Great. So these are what cardiomyocytes look like, and it's kind of fascinating. So this is a GIF from the Murano lab at the Max Delbruck Center for Molecular Medicine in Berlin. And you can see the heart is composed of these contractile cells, and somehow all of these cells need to coordinate their activity for the heart to act as a pump. So that's a fascinating thing on its own. Okay, so hopefully we're all keyed in on this concept of cardiomyocytes now, right? So why is it that after a heart attack, why is it bad? It leads to pathological remodeling, heart disease, heart failure, death. But why? And it turns out that adult mammals, which include me, a lot of you here, right? Adult humans, but also this adult mouse here, we lack generally the ability to regenerate our heart muscle or those cardiomyocytes after an injury like a heart attack. Okay, so to stress this point, let's say we took the heart out, we take a cross section, you see all this nice red staining. Let's see, these are all the live cells that are these cardiomyocytes that contract. So if this mouse, or even us humans, if we had a heart attack, we lose a lot of that red staining, and instead it becomes replaced by this purple staining, which is fibrosis or scar tissue. Not only that, you can see that the ventricle of the heart is dramatically enlarged, and also the walls of the heart are thin. So now you can kind of figure out or see why the heart has to behave differently or adapt to these pathological conditions. But the point is you see a lot of these the red staining is gone. So a lot of this heart muscle cell after a heart attack is gone. And generally in adult mammals, once this happened, it's gone forever. Okay, so adult mammals, after you lose your cardiomyocytes, they're gone forever. So they're very important. So take care of your hearts, right? What if I told you, though, is that some vertebrates actually, even as adults, possess the ability to regenerate their hearts? This is particularly true for one of my favorite model organisms, the zebrafish, right? So this was discovered back by Ken Paas in 2002. And essentially what he did was he took a pair of scissors and he cut a piece of the heart off, the apex of the zebrafish heart off. And over the course of a couple of months, he noticed that the heart was able to completely regenerate without scarring and fibrosis, what we'd normally see in an adult mammal, right? This also brings up an interesting question about the diversity of heart regenerative potential across the animal kingdom. And in a way, it makes us think that heart regenerative potential could be lost across evolution. So to drive this point home a little bit, I have a schematic here where on the x-axis, we have different types of animals, like different species, like zebrafish, amphibians, rodents, large mammals like pigs, non-human primates, humans. And on the y-axis, we have this qualitative description of cardiac regenerative capability, right? So in zebrafish, amphibians, reptiles, this tends to be higher. And then as we get towards mammals, it tends to drop off. Now, the problem is we don't know a lot of what's going on between amphibians and rodents because there's not a lot of model organisms where scientists can easily study the regenerative capacity in the heart. But there seems to be this trend where amphibians tend to have higher capacities, which drops off considerably towards the evolution of mammals. So this is from an evolution aspect, this is an interesting question to us. But what do animals like zebrafish and other vertebrates that can regenerate their heart specifically, or especially as adults, what can they teach us about heart regeneration? Specifically, like, what are the natural ways that the heart is able to regenerate? So I'm going to walk you through an experiment. So pay close attention. I'm going to have another question just to make sure that we're all on the same page here. So let's say we have the schematic of the heart. We talked about in the beginning question that the heart is actually composed of different types of cells. We agreed that the contractile cells in the heart are those cardiomyocytes, the heart muscle cells. That's depicted by these long rod-shaped cells here. Then let's say we have other types of cells in the heart, that these circular cells, right? So these could be the fibroblasts, it could be the endothelial cells, et cetera, et cetera. So let's say scientists came up with a genetic trick where if you add this drug, it's not important what it is at this point. It's just say it's a chemical trigger. In this case, it's tamoxifen. In the presence of tamoxifen, you're able to label all the pre-existing cardiomyocytes, these long rod-shaped cells in the heart, green. You make them express this protein called GFP. so that we can see it as green cells. So what does this kind of look like in the zebrafish heart? Here we go. So here's your zebrafish ventricle. All the pre-existing cardiomyocytes are green. And if you're interested in the genetics, this is the actual tools that they use, but we're not going to necessarily go into that right now. So the idea is you label all the pre-existing cardiomyocytes green, and then you take your pair of scissors and you amputate that apex off, right? You ask the question, what color does the newly regenerated tissue become? Okay, so that's the question that we're going to ask. And I'm going to give you the answer to what the color means, and I'm going to want you to try to interpret what that result tells you about how the heart regenerates. So the regenerated tissue is green, okay? So let's go to the next question. I'm going to set it up for you. So remember, we have a way to label all pre-existing cardiomyocytes green. We amputate the heart, and the regenerated tissue becomes green. Okay, so the question that I have for you this time is, from what cell type does newly regenerated heart tissue come from? It's a little bit tricky, but I think you guys have it. Ready? So the choices are cardiomyocytes, A, B, fibroblasts, C, endothelial cells, D, stem cells. Let's take about a minute for this one. You can think about it. I see what's going on. It's what I expect. So remember, pre-existing cardiomyocytes are green. The regenerated tissue is green. Where did the regenerated tissue come from? Take another 20 seconds or so on this. This is a tricky question, but it's a very important question to set up the basis of this talk. Okay, so let's wrap it up if you haven't. Okay, it's been about a minute. So let's end the poll right now. Let's see what people think. So I can share with you your results. I'll also drag them onto the screen. So let's say about half of you have said it's these cardiomyocytes, right? And then half of you have said stem cells, which makes sense because a lot of the time we think regenerating tissue, there's a bunch of stem cells in the tissue that become mobilized or activated in response to injury, creates new tissue. Makes sense. Okay, so I'm going to stop sharing. The correct answer is cardiomyocytes, right? Okay, the reason why is because you already labeled all the pre-existing cardiomyocytes green. All the cardiomyocytes that were there are green. The newly regenerated tissue is green. So the newly regenerated tissue had to come from the pre-existing cardiomyocytes. So the second most popular answer, stem cells. See, I predicted. See, I already had a circle planned for this presentation. A lot of the times we do think about stem cells, and this has been a point of controversy in the heart regeneration field for some time now. And currently, the model is that there are no cardiac stem cells in the heart. Okay. If it were stem cells, the regenerated tissue, you'd actually expect it to not be green. Because when you label the entire heart, it's only the cardiomyocytes that are labeled. So the stem cells wouldn't have been labeled during that pulse of that trigger. So this would not be green. Hopefully that makes some sense. But yeah, it's been debated. I think currently what people think is that heart regeneration does not rely on a stem cell population. That's the current understanding. So to drive home the key point, cardiomyocyte proliferation, the ability of the cardiomyocytes to divide, is important for the heart to regenerate, right? The green cells that were there need to make more copies of themselves in order to make the regenerated tissue, right? So this stresses the importance of cell cycle control. This is a topic that we're very interested in understanding, is how is the cell cycle, which is important for these cardiomyocytes to proliferate, how is it regulated? Because what these animals that can regenerate their hearts teach us is that those pre-existing cardiomyocytes need to enter the cell cycle again and divide to replace the missing tissues. So this seems to happen in animals like the zebrafish. It doesn't happen in the adult mammals in general, okay? So I kind of half told it, well, I'm not lying, but mammals do have a capacity to regenerate their hearts. But I've been stressing adult mammals for a while now, and this will lead me to the next point. So it turns out that while adult mammals have very limited capacity to regenerate their hearts after injury, baby animals, like this newborn mice, has a pretty strong capacity to regenerate their heart, right? This is the same animal now. They're only separated by developmental stages. So if you take the heart of a newborn mouse, take a pair of scissors, cut a piece of that off, over the course of about a month, that heart's going to be able to regenerate, right? With very little scarring and fibrosis. Again, very different than what you'd expect in an adult mammal. We can see what this looks like qualitatively across developmental time, right? So now on the x-axis, I have different developmental stages, like the fetus, when it's developing within its mother's womb, birth, when the animal is born, postnatal day 7, 1 week after birth, and then adult stages. So this is really cool. So initially, during fetal stages, cardiac regenerative capacity is very high. In rodents, right after birth, within the first 7 days of life, the animal loses its ability to regenerate. 7 days. That's really quick. So it goes from being able to regenerate its heart completely to not being able to regenerate within the first 7 days of life. This is another very interesting question that we are interested in pursuing. Developmentally, what is going on during this magical first week of life that makes this mammal lose its ability to regenerate? Okay. So we kind of calibrated on the cells in the heart, and that's important because that's where our focus is going to be. So remember, in order, what these animals like zebrafish taught us, and this is also true for neonatal rodents, is cardiomyocytes, the pre-existing cardiomyocytes need to be able to enter the cell cycle and divide in order to replace the missing tissue. So let's focus our attention again on those heart and muscle cells called cardiomyocytes. So during the regenerative phases where the heart can actually regenerate, this is our depiction of the cardiomyocyte. And we're going to get a little bit deeper into the cardiomyocyte. Specifically, I want you to focus on this cardiomyocyte. It has one nucleus. It's mononucleated. It has 2 copies of the genome, so it's diploid. Mononucleated diploid. 1 nuclei with 2 copies of the genome. So most mammalian cells will have 2 copies. It's 2N, or the ploidy of most mammalian cells has 2 copies of the genome. So check out what happens during this abrupt first week of life, right? Animal is born. Within a couple weeks of development, there's a sharp transition where the majority of these mononucleated diploid cardiomyocytes become binucleated, right? They're binucleated polyploid, so they actually have more than normal amounts of DNA in them, right? They have 4 copies of the genome, cardiomyocytes. So there's polyploidization that occurs, multinucleation occurs, right? So while the heart is regenerative, there seems to be more of these guys. After this transition, they become binucleated polyploid cardiomyocytes, and that correlates with the loss of heart regenerative capacity. Why is this? To help you see and understand how this process of binucleation and polyploidization occurs, let's take an overview of the cell cycle. So maybe you've heard about this before. So in the cell cycle, there's different phases. So there's G1, G2. These are resting phases. Don't worry about that so much. S phase is where DNA synthesis occurs. So when the cell needs to duplicate its DNA material, this occurs during S phase. And here in M phase, or mitosis, we'll see what happens. So let's walk through some of the steps in mitosis. but you have like interphase, prophase. Don't worry about these steps so much. I'm going to highlight the important steps. So one of the key things that occurs during mitosis is you can see initially there was one nucleus, right? Still one nucleus. Well, the nuclear envelope breaks down here, but now you can see at this step, you have segregation of the nuclei, right? So the nuclei, the nuclei divide. There's nuclear division during mitosis. During the last step of the cell cycle, cytokinesis, what you have is cytoplasmic division. So the stuff in orange or brown, that needs to be evenly distributed into the daughter cell. So this is what normally happens when a cell enters the cell cycle and tries to divide. There's a separation of the nuclei and then a separation of the cytoplasm. So let's look at our targeted cell type again. So this is binucleated cardiomyocyte. How did this arise? This is going to relate to our next question. So think about this diagram a little bit. We're going to do a little bit of troubleshooting. How did this arise? Binucleated cardiomyocyte. So the question I have for you right now, an error in which phase of the cell cycle results in binucleated cardiomyocytes? Your choices are A, G1, is the resting phase. B, S phase, DNA synthesis. M phase, mitosis, where the nuclei divide. Or D, cytokinesis, when the cytoplasm divides. So where along the cell cycle path was there a mistake that led to binucleated cardiomyocytes? Where do you think the error occurred? So you see what the result is. Where was the mistake? All right, so let's take another 20 seconds or so. You can chime in. Okay, let's wrap it in about 10 seconds. So if you want to chime in, please do so. Okay, so let's go ahead and end the poll. And let's share your results. So we have a large percentage of viewers that are saying M phase, mitosis, where there's nuclear division. And then a little bit more that are saying cytokinesis, where the cytoplasm needs to divide. Okay, so the correct answer here, so I'm going to stop sharing, is cytokinesis, when the cytoplasm divides. Because think about it. There are two nuclei, right? So the nuclei were able to divide, but the cytoplasm was not able to divide. So that's why you result with two nuclei in one cell. So if we go back to our diagram, where the block is here in this last step of the cell cycle, cytokinesis, right? So if we go back to this transition, what we think is going on is right after birth, those cardiomyocytes that are initially mononucleated and diploid, two copies of the genome, they undergo some transition. So they enter the cell cycle that one last time, and they fail cytokinesis, and the majority of them become binucleated. And at that point, what we think is these cells are locked up. They can't enter the cell cycle anymore. So in response to an injury in an adult stage, those cells cannot divide anymore. And that's why we've lost the ability to regenerate. But developmentally, where does this occur? In rodents, within the first week of life. Crazy, right? So fast. So I introduced two of these concepts to you. So first of all, that heart regenerative capacity may have been lost across evolution, but also heart regenerative capacity seems to be lost across development in mammals, right? Neonates we can learn a lot about how heart regeneration occurs by studying these neonates or babies. But as they transition to adults, we lose the ability to regenerate. So the question then becomes, well, is there some sort of common logic or link between this evolutionary effect that we're seeing and also developmental? So why don't we just start putting some of these pieces together so that we can see an overall view of how things look, okay? So we know about this transition. When the heart is able to regenerate, the majority of cardiomyocytes are mononucleated and diploid, one nucleus, two copies of the DNA. During the first week of life, there seems to be a transition where the majority, and when I say majority, I'll say 90% of rodent cardiomyocytes become binucleated, tetraploid or polyploid. So there's polyploidization, more DNA content in the cells. We talked about adult zebrafish. We talked about neonatal mice and adult mice. And just to think about the percentages of these mononucleated cells. So let's focus on these. Adult zebrafish, most of the cells are mononucleated diploid cardiomyocytes. Same thing is true for neonatal cardiomyocytes. About 100 neonatal rodents, about 100% are mononucleated cardiomyocytes. By the time this animal becomes an adult, this reduces down to 5 to 10%. Do the cardiomyocytes have the ability to proliferate? In zebrafish, they do. In neonates, they do. In adults, they don't. When tested for cardiac regenerative capacity, do these animals have the ability to regenerate? Adult zebrafish, they do. Neonates, they do. Adult mammals, no. So what this lets us see is there seems to be a correlation between animals that can retain a higher abundance of these mononucleated diploid cardiomyocytes and the ability to regenerate the heart. Okay, so that's kind of an idea that I'm trying to pitch right now to make sure that people see that when animals seem to be able to regenerate the heart, the cardiomyocytes tend to be mononucleated and diploid. If there's a transition or polyploidization where they become binucleated or just have more DNA content, so they arrest in the cell cycle, there's a block, then we've lost the ability to regenerate the heart. So there seems to be some correlation there. So this is important for us to understand what a little bit of this work that I had participated in or did as a postdoc in Guo Huang's lab at UCSF when I was doing my postdoc. So from that previous slide, what we can see is that the frequency of mononucleated diploid cardiomyocytes, we might be able to use that as a proxy, right? Or a sign or a, some evidence that maybe animals that have higher percentage populations of these cells might be able to regenerate, right? So we're going to use it as a proxy for heart regeneration, the frequency of these mononucleated diploid cardiomyocytes in the heart. So what we did when I was a postdoc in Guo Huang's lab, alongside my colleague Kentaro Hirose, is we took animals such as the zebrafish and the mouse, like animals we just talked about, and we'll take their heart samples. Now, what we can then do is dissociate the heart into cells, and particularly look at the cardiomyocytes. These are the long rod-shaped cells. So by doing experiments like these, we can quantify and see, oh, okay, this is a mononucleated diploid cardiomyocyte. These are binucleated cardiomyocytes, right? So you can take images and calculate, okay, in these animals, what's the percentage of cardiomyocytes that are these versus these? So we can do that for model organisms like the zebrafish and the mouse, but we can also do that for a bunch of different other animals too. And that's exactly what we ended up doing. So not only did we survey conventional model organisms like the zebrafish and the mouse, but we also had heart samples from bats, armadillos, platypus, naked mole rat. That's kind of a fun animal nowadays. The gray whale, bow-headed whales, tenoraks, basically trying to fill in that evolutionary void that we've been missing because we've only been focusing on model organisms, right? So we're able to calculate what's the percentage of these diploid mononucleated cardiomyocytes in each of these animals. And we can arrange that on a graph like this, right? So in total, what we did was we analyzed cardiomyocyte nucleation across 41 different species. 6 of them are non-mammals, 35 of them are mammals. And for example, if you want to just see in mice, cardiomyocytes look very different, right? In adult, these are all adult animals, by the way, but in mice, you can see the cardiomyocytes tend to be more rectangular. In the platypus, they tend to be longer. In zebrafish, they kind of look like worms. So it's kind of fascinating on its own. Like you think of a cardiomyocyte as a cardiomyocyte, and all cardiomyocytes are the same, but it's not true. If you look at the cardiomyocytes in all these different animals, even at the individual cell level, they look different, right? So when we calculate the ploidy of these cardiomyocytes, we find that, as we would expect, mice has about 10% mononucleated diploid cardiomyocytes left as adults. The majority became binucleated. They've exited the cell cycle and stuck. But what we did find is a bunch of animals that you would consider mammals that retain quite a high number or percentage of these mononucleated diploid cardiomyocytes. So if you put that on this table that we've been forming thus far, what these results suggest that there might actually be some mammals that retain at least enhanced ability to regenerate their heart. But this remains to be tested, and it's hard to test this, especially in non-model organisms, right? So that's an interesting observation. So oftentimes, like science will give you what you're testing for, but you also have to have appreciation that the number of animals in the world are much more diverse than, say, a mouse and a fish. So what we learn from these animals may only apply to these. So we have to kind of sometimes broaden our outlook on things. Okay, so anyways, we ended up collecting all this data, surveying across all of these different animals, and we tried to figure out, okay, is there a trend somewhere? Is there something that's predictive to say if an animal is going to have higher diploid cardiomyocyte abundance than another animal, right? So we looked at a bunch of things, and I'm going to make this quick, like body weight, heart rate, blood pressure, and we don't see any correlation whatsoever. But we did see something interesting, and I want you guys to be able to interpret this. So like as scientists, what we have to do is interpret data. So I'm going to show you the data, but let's look at this interrelationship between another factor that we were looking at, which is metabolism, okay? So let's take a look at the interrelationship between animal metabolism and cardiomyocyte ploidy. So this is the data. I'm going to introduce you to the data and draw your attention to just several key things and where they are, but I want you to think about where the data points are, right? So on the x-axis, we have standard metabolic rate. So you can think about it a little more simplistically as like animals that have higher metabolism are on this side. So it's increasing metabolism this way across the x-axis. And on the y-axis, you have the percentage of diploid mononucleated cardiomyocytes. So animals that have more of that mononucleated diploid cardiomyocyte that we're using as a proxy of heart regenerative capacity. So animals so like here on the y-axis is more diploid mononucleated cardiomyocytes going up. So increasing 0 to 100%, and then standard metabolic rate going across the x-axis. So look at this data and think about where the data points live and what we can interpret from it. So this is your next question, okay? Just to remind you, here's the data so you can use it as reference. So think about this. What is the relationship between animal metabolism and the percentage of diploid cardiomyocytes? So your choices would be A, animals that have lower metabolism have less diploid cardiomyocytes. Animals have lower metabolism have more diploid cardiomyocytes. Animals that have higher metabolism have less diploid cardiomyocytes, or higher metabolism, more diploid cardiomyocytes. So look at the data itself. Sometimes that's an important skill, right? We're looking at data, we're trying to extract information from it. What does the data tell you? Look at the points on the graph. How do you interpret this data? So let's do another 30 seconds. This is a hard one. Like sometimes it's hard to look at a graph and extract information from it, but it's an important skill, at least to try, right? And there's no right or wrong. Well, I mean, it's important to try, right? Let's keep it going let's do another 10 seconds here. I still see people chiming in. All right, maybe let's try to wrap it up in about 15, if possible. Okay, maybe 5 seconds. All right, so let's end the poll now. And let's share the results. So you guys should be able to see what you collectively said. You're interpreting data now, right? and I'll show you on my screen too. But the majority of you said animals that have lower metabolism tend to have more diploid cardiomyocytes, okay? And that's actually the correct answer. So good job there. And let's walk through that a little bit. So I'm going to stop sharing this. So you can see if metabolism is increasing along the x-axis and the percentage of diploid cardiomyocytes is increasing across, as you go up on the y-axis, then animals that have low metabolism, right, tend to have higher diploid cardiomyocytes, because look at the spread of all of the different points. So it's an inverse correlation. Conversely, animals that tend to have higher standard metabolic rates have lower diploid cardiomyocyte percentage. So there seems to be a trend there, okay? And the other thing I want to point out is by classification, there seems to be a transition occurring where these animals are ectotherm or cold-blooded animals. And as you move this way, they end up being endotherm or warm-blooded animals. So there might be some transition there. So pay attention to that. Low metabolism, high diploid cardiomyocytes, cold-blooded. High metabolism, low diploid cardiomyocytes, warm-blooded, right? So we found this inverse correlation between mononucleated diploid cardiomyocytes and standard metabolic rate. So what we wanted to think about next is which pathways do we know about that regulate animal metabolism? So cutting or like shortening the story a bit, we ended up focusing on thyroid hormones, because thyroid hormones are classical regulators of metabolism. Thyroid hormones are secreted from the thyroid gland as T3 or triiodothyronine or thyroxine. So thyroxine is a prohormone and usually needs to be converted to T3, to the more active state. And they're well known to increase metabolism, but also maturation of various tissues in the body. Its role in metabolism is specifically shown here, where, for example, if you take examples from eutherians like rabbits, or marsupials like this bandicoot, or monotremes, or the egg-laying mammals like the echidna, what you see is if you perform thyroidectomy experiments, so you remove the thyroids, so that would deplete the system of thyroid hormones, you get a drop in the standard metabolism of these animals, but specifically eutherians and marsupials. So for whatever reason in monotremes, it doesn't seem to change much. So it could be that in these mammals, eutherian marsupials, there may have been an evolution of thyroid hormone signaling to support that elevated metabolism, right? Does this apply to this framework that we've built so far? And it kind of does. And this is what made us kind of pursue thyroid hormone as a possible signaling mechanism that could be driving this binucleation, loss of heart regeneration across birth. When we measured thyroid hormones during that first week of life, so on the x-axis, you have postnatal days, 0 to 7. And then here's T4, or the prohormone thyroxine. We see an increase in that. Also with the triiodothyronine, we also see an increase there. So it seems like across development, thyroid hormone levels in circulation are increasing, right? So it made us wonder, like, okay, so maybe this increase in thyroid hormones, it's driving animal metabolism during this postnatal phase, and that's driving itself. That's driving the loss of heart regenerative capacity in these rodents, all right? So how do we test that? So that's the next question. How can we test if thyroid hormones regulate cardiomyocyte proliferation? Well, thyroid hormones are nuclear receptors, right? So they live in the nucleus already and they bind to thyroid hormone response elements. So this is this TR, that receptor. And in the presence of thyroid hormone, like triiodothyronine or T3, this thyroid hormone receptor can recruit activators to turn on target genes that are downstream thyroid hormone signaling. The protein products of those genes are going to be what facilitates metabolism or promotes maturation, et cetera, et cetera. So the strategy that we use, to simplify it, is we're using a genetic trick in mice to overexpress a dominant negative form of this thyroid hormone receptor. What does that mean? we're forcing expression of like this mutant version of the thyroid hormone receptor that can't recruit these transcriptional activators. It's serving as a dominant negative because it outcompetes the endogenous receptors that normally sit out here, which can recruit the transcriptional activator. So we're basically flooding the cell with this thyroid hormone receptor that can't recruit transcriptional activators. So the downstream signaling doesn't happen. And we do this specifically in cardiomyocytes. So in mouse, there's the power of mouse genetics is that you can have tissue-specific or cell type specific genetic perturbations, right? So I'm not going to go too deep into that, but basically we're blocking cardiomyocytes specifically from responding to thyroid hormone by expressing this dominant negative version of the thyroid hormone receptor that can't recruit transcriptional activators. Okay, so what happens when we generate these animals? So at postnatal day 14, so this is 2 weeks after birth, in black are control animals. These are the genotypes. We can talk about that if you're interested, but basically for this, black is control, red are animals where the cardiomyocytes, CMs, can't respond to thyroid hormone TH. What do we see? The animals that are mutant for thyroid hormone receptor, the hearts end up bigger, right? So here's body weight, doesn't change too much. The heart weight increases, and so does the heart weight body weight ratio. So these animals where you're blocking thyroid hormone signal, the hearts end up being bigger. Okay, let's look at the cardiomyocytes using the same assay as before. We take hearts from these animals and calculate the percentage of cells that are mononucleated diploid, right? The regenerative cardiomyocyte. What we see is that there's an increase in cardiomyocyte number in these hearts. There's actually an elevation in the percentage of diploid cardiomyocytes. So more of the cardiomyocytes that we would expect to be pro-regenerative. And the cardiomyocytes end up being a little smaller, okay? If we take sections of these hearts and we stain for PCM1, which labels cardiomyocyte-specific nuclei, so all of these green dots represent the nuclei of the cardiomyocytes, and Ki67, which is a marker of cardiomyocytes that are inside of the cell cycle, so they're actually actively in the cell cycle, we see in these thyroid hormone mutants, there's an elevation in the number of cardiomyocytes in the heart that are positive for this cell cycle marker. So not only do we see more cardiomyocytes, they're less binucleated, and the majority of them are still in the cell cycle. Oh, not the majority, but there's a large percentage of them that are in the cell cycle. So developmentally, it looks like the cardiomyocytes are not exiting the cell cycle during that first week of life as they would normally. So we wanted to test then is in these thyroid hormone mutants, if we do a heart injury to that heart, so in these adult animals now, in this case is ischemia reperfusion, so we temporarily ligate a suture around the left anterior descending coronary artery, so that will temporarily block blood flow to this region, but cause some damage. We find that these animals that are mutant for thyroid hormone, also that the sites of the tissue that are closest to the infarction or that area of damage tend to also be more proliferative, more so than controls. So you can see this is the border zone, so the region closest to the injury, there's more Ki67 cell cycle marker positive cardiomyocytes, right? Not only that, if you look at scarring in these thyroid hormone tissues, so these mice can't respond to thyroid hormone, we see less fibrotic area in the sections that we look at. And more importantly, if you look at the function of the heart, so we use echocardiography and look at ejection fraction, in animals that are mutant for the thyroid hormone receptor, so the cardiomyocytes can't respond, while the controls tend to have a declining effect in function, thyroid hormone animals mutant animals are able to recover a bit, suggesting that thyroid hormone mutant animals have increased capacity to regenerate. So what's the model that we think is going on thus far? So thyroid hormones we think are important for increasing animal metabolism and might be important for driving this transition between ectotherm or cold-blooded animals to warm-blooded animals. At the same time, in parallel, there seems to be a transition that occurs where these animals have more mononucleated diploid cardiomyocytes, those pro-regenerative cardiomyocytes, where now, as you increase in thyroid hormone levels, you're starting to promote more binucleation, cell cycle arrest. And this correlates with the loss of cardiomyocyte proliferation and regenerative capacity. So the model is elevation in thyroid hormone levels increases energy metabolism, but decreases cardiac regenerative potential as a trade-off. And the question then becomes why? So I want to throw this out there, but in 1969, Richard Goss, who was a regenerative biologist, proposed that it seems that regeneration is not necessarily adaptive, but that it has disappeared or persisted as other more important physiological attributes evolved with, which it may or may not have been incompatible, right? Hence, before any intelligent attempt can be made to restore regeneration where it does not normally take place, it will first be necessary to learn what physiological advantages took precedence over regeneration in the course of natural selection. So here's what I have to leave you with this part of the story. And then I have just a short story afterwards, or just to wrap it up, what we're more looking at currently. Is this trait of being warm-blooded, which requires elevated amounts of metabolism, right? You need to be able to regulate your body temperature. Even if it's cold outside, your body needs to be at a set temperature that takes energy, right? It needs to increase metabolism. Is that more important for mammals than being able to regenerate your heart? So is thyroid hormone one of these links that are important for us to become endotherms, but it's incompatible with heart regeneration? It actually drives the cardiomyocytes out of the cell cycle and causes us to lose regenerative capacity, okay? So that's something I want you to think about. Is there a trade-off in being warm-blooded versus being able to regenerate your heart? Okay. So we have, I don't want to go too long because I definitely want to answer a few more questions, or I have time for questions, but I just want to leave you, let's continue with this. So the question is thyroid hormone the only pathway that controls cardiomyocyte proliferation and ploidy? And I want you to focus on this particular piece of data, okay? So this is a quick question, number 5. And then again, this is an exercise in looking at data. If you saw, here's your control animals, here's your thyroid hormone mutants. This is the percentage of diploid cardiomyocytes. Question number 5 would be true or false. Thyroid hormone signaling is the only pathway that drives cardiomyocyte binucleation. Is that true? Pay attention to the data itself. Look at the y-axis. Particularly, that's my hint to you. Let's do another 30 seconds or so. Look at the y-axis. Let's wrap up in about 15. Okay, so let's stop the poll here. So I think the majority of you said false, right? Here's the result. And the correct answer is false. And why is that? Okay, so let's click that away. Because when you look at this data, even with thyroid hormone depleted, the cardiomyocytes can't respond to thyroid hormones. Look, you only have 30% of the cardiomyocytes that remain diploid mononucleated, that regenerative type of cardiomyocytes. That means 70% of the cardiomyocytes are still becoming binucleated. So it means there must be some other pathways. Okay, so I'm going to go through this part relatively quickly, just to consider time. But here's some other pathways that we're interested in looking at. So what other pathways work with thyroid hormone signaling to control cardiomyocyte proliferation after birth? You can think of birth, there's a bunch of things happening, right? We focused on plasma hormones, like thyroid hormone, but another thing we were focusing on was sympathetic nerve activity. the major thing I want you to think about sympathetic nerves. So these are the things that are important when your fight or flight response, right? When you start to panic, your heart starts to beat faster. You need more metabolism, right? You need more energy. A lot of that is driven by sympathetic nerves. But what made us look at the sympathetic nerve activities, particularly its role in regulating brown fat mediated thermogenesis. So along this line of endothermy or warm bloodedness. So brown fat is like kind of a mammalian innovation that allows us to uncouple products from the respiratory transport. So instead of producing ATP, instead the mitochondria can produce heat. So this allows us, as one of the mechanisms that we can use to maintain our own body temperatures, even when the environment is cold outside. So what we wanted to see, okay, so sympathetic nerves typically are activated by neurotransmitters like norepinephrine, and they signal through alpha and beta adrenergic receptors. So essentially what we wanted to see if there's an interaction between thyroid hormone and sympathetic nerve activity. And we took a more drug approach, right? So we use these inhibitors against these adrenergic receptors that sympathetic nerve signal through. So we block alpha adrenergic receptors with a drug, we block beta adrenergic receptors with a drug, and then we block thyroid hormone with a drug. And we do this in combination to see if there's any combined effects, particularly on cardiomyocyte, cell cycle activity, and body temperature. I was going to ask a question, but I think in the interest of time, I'm just going to go through some of the key results. In combination, when you block all of these pathways together, thyroid hormone plus the sympathetic nerve signaling pathway, basically through adrenergic receptors, the body temperature of the animals drops considerably. If you look at the trace through the first 2 weeks of life, animals that are injected with these drugs can't maintain their body temperatures at the 2 weeks, whereas the body temperatures of normal animals are 37 degrees. So this idea of like warm-blooded to, or cold-blooded to warm-blooded transition. Let's skip this question for now, but basically the last question, which I'm not going to ask for the interest of time, just has you think about like this trends that we're talking about, increasing metabolism, more diploid cardiomyocytes. What would you predict would happen if you block thyroid hormone and adrenergic receptor signaling if it drops the body temperature of the animals, right? The correct answer, what we ended up seeing, is that there was even a further increase in cardiomyocyte proliferation. So using the same assays before, we can see that more cardiomyocytes are in the cell cycle, even more so than all of these other controls. So control, just blocking adrenergic receptor signaling, thyroid hormone alone, but the red is higher than everything else. So when you block these two pathways together, there seems to almost be some combined effect to increase the cell cycle activity of the cardiomyocytes in the heart. There seems to be more diploid cardiomyocytes, right? If you even do an injury model on these animals, there seems to be an increase in heart regeneration or increase in function, whereas all these other conditions drop. So there's something about blocking adrenergic receptor signaling and thyroid hormone signaling together that we think might even further enhance the ability of the animals to regenerate by blocking that shutdown of cardiomyocyte cell cycle arrest, right? In these animals, we also see, less scarring. So I'm just kind of going through this quickly, right? So the current challenge that we have, and what I'm currently studying at San Jose State, is we don't really know the mechanisms that are downstream all of these different pathways. And because animal physiology is pretty complicated, right? We take these animals, we inject them with all these drugs, but we're focusing on the heart. But physiology is a very integrative type of field, right? The heart, cardiovascular system influences other systems influences the heart. So we don't really know if what we're seeing when we're doing these drug studies is direct or indirect action, the mechanisms, direct or indirect. So at San Jose State, what we've been focusing on is on in vitro models to culture the cardiomyocytes directly from the heart in a dish, and then treating them and activating these pathways singly or in combination to determine the direct effects of these pathways on the cardiomyocytes. We do a bunch of things, like we look at we're taking advantage of this new technology. It's called like digital holographic imaging to look at how cardiomyocytes divide in culture. So you can see a cardiomyocyte here. This was able to successfully divide, or perhaps binucleate. So here's a cardiomyocyte that tries to enter the cell cycle, divides, but ends up becoming binucleated and snapping back together. So we're trying to understand this on the cellular level, but at the same time, we're also trying to understand the genes that are involved downstream of these pathways using these in vitro assays. Okay, so at the end, so just to summarize, because it's 6 o'clock, I want to make sure there's time for questions. What we're very interested in is what's causing cell cycle arrest in mammals when you start from a newborn and become an adult. We're focusing on these neurohormonal signals such as thyroid hormone or sympathetic nerve activity, or those neurotransmitters, adrenergic signaling. And we want to understand is like, how does this cardiomyocyte, this mononucleated diploid at birth, why is it getting locked up and becoming binucleated or being able to divide? What is the logic behind that? Because I think if we figure out what signals shut down the ability of these cardiomyocytes to divide after birth, we may one day be able to unlock the ability of these adult cardiomyocytes to regenerate. So we've got to kind of figure out what's shutting them down first during development. Then we can think about how we might be able to unlock that. Like, what are the roadblocks for these cardiomyocytes to be able to divide? Once we identify that, we might be able to unlock that in adult animals, and then, you know, revive the ability of the heart to regenerate in these adult mammals. Okay, so with that, I just want to acknowledge a few people. So this is the Huang lab at UCSF. This is where I did my postdoc. And then the two papers that we talked about, so with Kintaro, this is with the thyroid hormone mutant studies. And then with Shi, these are with this combined drug studies. So they've always been very supportive. This is Gua Huang, he's super supportive as well. But this is my lab currently at SGSU. We have graduate students as well as undergrads. They're a fun bunch. And then funding support from C-Superb, internal grants from San Jose State University. And then we recently obtained an R16 from NIH. So our funding, our research is also supported by the NIH. Okay, so the last thing I want to say is just to thank you, just a big thank you to everybody here at Café Sci and the moderators. And it's a great opportunity for me. So hopefully, you know, we can have continued discussion about this. But at this point, I'm opening up for questions if anybody has any.
Great. Thanks, Alex. This was awesome. Yes, as Alex said, please use the Q&A button at the bottom to ask your questions. I see we already have a few in here. So I guess we'll go ahead and jump right in. So we got the first one here, says, can the diploid cardiomyocytes be produced from stem cells?
So that's actually an important, so there's different strategies to try to get the heart to regenerate. A lot of our research right now is focused on trying to unlock the natural ability of the heart to regenerate. We learn those mechanisms from animals like zebrafish, neonatal mice, right, that can regenerate their hearts. What this question is asking is, can we make diploid cardiomyocytes from stem cells? And that's another approach as well, right? So we can take, for example, in humans, we can take human-induced pluripotent stem cells and differentiate them to cardiomyocytes. And yeah, they will become diploid mononucleated, right? The problem with that approach so far has been, I think, an issue with maturation. Because remember, cardiomyocytes, what they look like as newborn, when you're a neonatal mouse, look very different than an adult. But a lot of the times they're trying to transplant cells into the adult. So with stem cell based approaches, and there's a lot of interest in this field too, is that we can only push the cardiomyocytes to kind of look fetal or neonatal. We can't really make them like an adult. So there's a lot of issues with the transplantation studies and them actually taking. So you can make diploid mononucleated cardiomyocytes using stem cell based strategies, but they might not be in a compatible maturation or developmental state to be able to graft into an adult heart. But that's a different method of trying to regenerate the heart. Yeah.
Interesting.
Great question.
Yeah, great question. We got another one here, a bit longer. So here we go. It says, given what you said about thyroid hormones, what impact might taking Synthroid or its generic alternative have on a human being who's having heart issues? What if they take that medicine for thyroid condition? Can dealing with a thyroid condition contribute to heart issues?
So I think, so if we're manipulating thyroid hormone signaling with drugs after development, that's something we're not really so sure about. Because if you pay attention to what I want to direct your attention to is like we've been inhibiting thyroid hormone signaling in the cardiomyocytes from birth or earlier than birth, and then grew those animals up to adulthood. So if you repress thyroid hormone activity in the cardiomyocytes from birth, maybe the heart can have an enhanced regenerative potential. But just manipulating it like after birth, I don't know how plastic, like once the heart starts, so thyroid hormone is a maturation hormone. So it's going to start trying to mature the heart, mature the cardiomyocytes. And I don't know how plastic that is. Like, can we, for example, inhibit thyroid hormone later and then recover more regenerative capacity? I think we were trying to, when I was a postdoc, we were trying to do those experiments, but we don't know that yet. So I don't know.
Awesome. We have another question here. It says, were you to be successful in unlocking the adult cardiomyocytes, would this be associated with an increased risk of malignancy?
That's an important question. I think that's true for a lot of even stem cell based approach. When you're trying to manipulate cell cycle, there's always a chance that things can become cancerous. So, I don't know, like if we were able to somehow allow cardiomyocytes to enter the cell cycle and be able to divide, yes, that's going to be an important thing to try to control. Like, how do you not get over proliferation, just enough to regenerate the function of the heart, but still, keep control of the system?
Yeah.
Yeah, that will have to be a thing to make sure if that ever comes to play. But those are true also for a lot of stem cell based approaches. So maybe there's a way to temporarily just turn on or fix or increase the proliferative capacity of the cardiomyocytes and then turn it off again. Yeah, that would be something to think. But naturally, the heart does not, you don't ever really hear about heart cancer so much, right?
Yeah.
And that's because those cardiomyocytes are not very proliferative. Because we lost that ability somewhere during that transition through birth or from early stages to adulthood. So we don't hear a lot about heart cancer normally, but now when we're starting to mess with their cell cycle activity, and maybe it's something we should think about.
Yeah, no, these have been really great questions already. Another question here says, can newborn humans regenerate their hearts at all?
I think there's like ethical issues with formally testing that, but there have been like case studies like clinically where like a human baby were born with some heart issue that caused damage, and at least it seems like they were able to grow up without any. So like with case studies are smaller and maybe it's possible. I would also say, like, in rodents, the transition to lose heart regenerative capacity happens really quick, right? Over the course of a week, 2 weeks. But in humans, the progression through polyploidization and cardiomyocyte cell cycle exit happens over a course of years. So the timing of it in humans is a little bit different. But there are some case studies that suggest that a newborn is able to recover from like a cardiac injury at birth. But you can't formally test that for a lot of different reasons.
Makes sense. Another question here, it says, does exposure to fetal blood affect the ability of adult cardiomyocytes to regenerate?
That's an interesting question, and it could. So we've been thinking about it in terms of factors in the blood that increase during development that force things out of the cell cycle. What this question is kind of thinking about, and which we haven't really thought about, is that are there factors in the blood during those cardiac regenerative stages that promote it, right? So I haven't tested that myself, but I don't recall either. But then that would suggest that maybe there are actually things in the serum that promote heart regeneration. But that's a good question too.
Great. Another question here says, are there any animals other than zebrafish and rodents in which heart regeneration has been documented either throughout their lives or as newborns?
So I think zebrafish has been like kind of like, because of the early work of Ken Poss, it's attracted a lot of attention as a heart regenerative model. Like I mentioned, I think there's a lack of model organisms in general where we can really survey the natural heart regenerative capacity. I think people are starting to do that work now. So people, for example, we had data in there that suggested that naked mole rats, maybe people have heard about those guys, they're really crazy, and they do have an elevated percentage of those diploid mononucleated cardiomyocytes that we think could be pro-regenerative. There's been some studies testing whether or not they are able to regenerate the heart. So there's a pretty recent, so we'll just see how that actually ends up looking up. Another mammalian model that seems to be attracting a lot of attention lately is the Spiny Mouse. Have you heard of that? Maybe?
Nope. I mean,
so yeah, they're pretty cool. So you could look at like the desert spiny mice, but they have like this crazy capacity to like regenerate huge amounts of their skin scar-free. So as a mammalian model for regeneration, that's an upcoming thing. And I think people have been trying to test whether or not, there's been several studies, I think, published within several months, within a few months time span, that are trying to test with. So I think basically the question is, I think we've learned a lot from these model organisms, neonatal mice, adult mice, rats, zebrafish. I think scientists are starting to venture out and try to formally test the ability of regeneration to occur in these other animals as well. But what we know mostly is from the model organisms.
Gotcha. This next question is sort of related to, we were just talking about this, but I thought it was funny. It says, can zebrafish get heart cancer?
I don't know. I don't know, but there must be some mechanism. I don't know if zebrafish honestly can get heart cancer, but their hearts remain proliferative throughout their entire lifespan, even as adults. So there might be mechanisms. Like there must be some. They must know, they have the right amount of breaks to know like how much to tune proliferation of the cardiomyocytes up and then shut it back down. But I honestly don't know if they can get heart cancer.
Yeah, that would be an interesting one to study. Another one here says, is it possible to either selectively or non-selectively inhibit the thyroid hormones directly without affecting the receptors and thus fix the cytokinesis step in human cardiomyocytes so as to heal injured tissue due to cardiac trauma, such as heart attacks, ideally without increasing the risk of carcinogenesis?
So is it possible to selectively or non-selectively inhibit thyroid hormones direct? So I think the problem is that, thyroid hormones don't just affect the heart. They'll affect everything or a lot of different tissues. So I think if you mess up the thyroid hormones themselves, you might have some profound effects on the entire animal. And that's some of the reasons why like those drug studies that I talked about briefly at the end are complicated interpreting, because you're inhibiting. So basically we treat the animals with PTU, phenylthiouracil, which blocks production of thyroid hormone. But it's going to affect all the tissues.
Yeah.
So I think that's the important part to stress with regard to that question, is that a lot of tissues rely on thyroid hormone for their maturation and development and possibly function too, right? So if you mess with the thyroid hormones themselves rather than the receptors in a tissue specific way, you might have some pleiotropic effects or something.
Yeah. Great. question, this question is relating to a previous question earlier on. It says, is there any other way to stimulate complete cell division in cardiomyocytes that have failed the cytokinesis step?
That would be great. I think that's what we're trying to figure out. So at least in vitro, there's been some studies that show that binucleated cardiomyocytes can actually divide. It's weird, like they have more centrosomes than normal. So I think in vitro, there's evidence that those binucleated cardiomyocytes may be able to divide. Whether that's true in vivo or in the animal remains to be seen. But I think that would be the holy grail, would be that's what people are trying to figure out. Can we get the cardiomyocytes that are already binucleated to divide? So some ways that people try to solve that is, well, just overexpress those cell cycle proteins and make them divide. So forcing the cell to enter the cell cycle is one thing. I think the field has made good progress in figuring out factors to make them enter the cell cycle. And I think relating to that question, I think where the gap is right now is we don't know how to make them complete the cell cycle and make two cells. Like oftentimes you can make a polyploid cardiomyocyte enter the cell cycle, but they just end up becoming even more polyploid, like more nuclei or more, like they can't divide. So I think that's the real big question. Like if you think about from the point of view of trying to make heart regeneration occur by making cardiomyocytes divide, we can get the cells to enter into the cell cycle, but completing it, especially when they're already binucleated or already locked up, I think that is where the current question is. We can get them to enter, but not necessarily complete.
Yeah, that's awesome. Couple more questions here. It says, would we want to control the regenerative capacity only as a treatment in response to a specific injury, as opposed to having this ability turned on inherently at all times so that you would avoid any unintended consequences?
So it's basically, what's the goal here? Do we want to make like heart regeneration like a 24/7 type of, like if we can unlock heart regenerative capacity, do we want to have it unlocked 24/7? Or do we want to have it unlocked only after an injury? I don't even know. I don't necessarily think we're even at that stage yet, but I would think is we would want something more controlled.
Yeah.
Because there are those cases that we don't know. Like if you're messing with cell cycle of cells, it can give rise to like cancer, and you don't want that. So from a therapeutic point of view, I think the more control we can have over the process, whether it be through unlocking natural cardiac regeneration or transplanting stem cells or whatever it is, I think we do want to clarify, like this is treatment, and then after that we want things to be normal again.
Yeah.
Right?
That makes sense.
Yeah,
definitely. Coming up on the last few questions here, one says, would it be possible to develop an in vitro process of regeneration over a scaffold using cardiomyocytes? What advantages, if any, would this route have over a stem cell route?
So that's another approach in regenerative medicine, because and that's something we don't really know too much in the field. So when you say scaffold, we're talking about like extracellular matrix. So a lot of the times when we think about cells, you see it in a textbook, like, oh, it's a cell. But that cell's not floating around in space. It's actually embedded in extracellular matrix. And I think that's an active area of research too, is like how much does the extracellular matrix and the signals that are embedded within the matrix, how does that regulate cardiomyocyte proliferation and regeneration? So I think, yeah, at least we can learn more if we can model extracellular matrix, for example, from fetal animals versus adults. I think that's another ongoing area of research, like what elements, non-cellular elements that are embedded in the matrix, how does that impact cardiomyocyte proliferation or the ability to divide, like the stiffness, things like like mechanotransduction stiffness, or it's very complex. So I've been very focused on just the cardiomyocytes, how do they divide, but in vivo, like cells are embedded in matrix to form the organ. The organ works as part of an organ system and multiple organs. It's very complicated.
Yeah.
Right. But
Yeah.
that's a good question. I think that's another thinking about scaffolds, especially what's like, how does the matrix change over development is interesting. Yeah.
Yeah, no, these have been all fantastic questions and really demonstrate how engaged the audience is. So this is awesome. I think we'll close with this question and wrap up. So this is the last one here. It says, could the cancellation of cytokinesis in the cardiomyocytes have the unintended benefit of preventing cardiac sarcoma?
Well, that's what I'm saying. I think like there must be a reason that for whatever reason in mammals, the cardiomyocytes exit the cell cycle and undergo polyploidization. And we don't know why, right? It could be preventative for certain conditions, like maybe it's protective, right? So like, why does a cardiomyocyte have to, for a mouse, for example, why does a cardiomyocyte have to have 2 nuclei? Why does it need to exit the cell cycle? Is it protective for certain diseases? Is it protective for certain states? I don't think we know that.
Yeah.
Is it important for function? Maybe people say like, oh, more DNA content. Maybe we can like transcribe more protein to support contractility more. I don't know. I don't think that's necessarily, I guess fundamentally, we just don't know why they even have to undergo polyploidization, or what's the real function of, or the benefit of ploidy or increased ploidy in the cardiomyocytes. So it could be to prevent certain diseases, or
Yeah. No,
I don't know. Yeah, I mean, like the idea that neonatal mice can regenerate. I forgot to quote the actual. So that was discovered by Enzo Perello and Eric Olson's lab in 2011. So it's kind of like this idea of studying the neonatal mouse and this transition. The transition has been studied in the past, but the fact that neonatal mice can regenerate their hearts hasn't really received a lot of. I think it's very interesting. Like people are very interested in now, but it's since 2011, so it's only been 10 years. But I think there's a lot that we don't really know yet.
Yeah. Yeah, it sounds like a lot more research will go a long way here in the next few years, right?
Yeah, and see which strategy is going to come up on that's going to actually be a benefit. Is it reviving the ability of cardiomyocytes to enter the cell cycle and divide? Is it going to be a stem cell based approach? There's actually other types of approaches, and we don't have to have that discussion here, but I encourage you to look, and there's a lot of different parallel approaches that people are trying to do to recover the heart, because it's like, again, 1 in 4 deaths in the United States.
Yeah.
Heart disease. So like it's from a financial standpoint, economic standpoint, there's a lot of benefit to being able to help patients.
Yeah. No, absolutely.